In the name of Allah, the most beneficent and the most merciful. Mehwish Khan is here to present you the topic gluconeogenesis. Dear students, hope you are well and doing great. The video on the topic gluconeogenesis contains instructive and educational content that is both shareable and easy to retain. So let's start. What is gluconeogenesis? Gluco stands for glucose, neo stands for new, and genesis stands for synthesis. So it is the formation of new glucose molecule from non-carbohydrate precursor occur in the liver. For example, these precursors are lactate, pyruvate, glycerol, and certain alpha keto acids. Sometimes kidney can also make new glucose in the form of metabolic acidosis or starvation. Gluconeogenic pathway. Gluconeogenesis is the reverse of glycolysis. Three glycolytic reactions that are catalyzed by hexokinase PFK1 and pyruvate kinase are irreversible. In this pathway, alternate reactions catalyzed by different enzymes are used to bypass these obstacles. Gluconeogenic pathway and its relationship to glycolysis. First of all, glucose is converted into glucose 6-phosphate in the presence of hexokinase, and glucose 6-phosphate is converted into fructose 6-phosphate in the presence of phosphohexose isomerase. And fructose this 6-phosphate is converted into fructose 1,6-bisphosphate in the presence of phosphofructose kinase 1, and this fructose 1,6-bisphosphate is converted into dehydroxyacetone phosphate in the presence of aldolase, and this dehydroxyacetone phosphate is converted into glyceraldehyde 3-phosphate in the presence of triose phosphate isomerase. This glyceraldehyde 3-phosphate is converted into 1,3-bisphosphoglycerate in the presence of glyceraldehyde 3-phosphate dehydrogenase, and 1,3-bisphosphoglycerate is converted into 2-phosphoglycerate in the presence of phosphoglycerate kinase, and this 3-phosphoglycerate is converted into 2-phosphoglycerate after the action of phosphoglycerate mutase, and 2-phosphoglycerate is converted into phosphophenol pyruvate in the presence of enolase. And the last step is the phosphophenol pyruvate is converted into pyruvate after the action of pyruvate kinase. Synthesis of PEP. To synthesize PEP, we require two enzymes that are pyruvate carboxylase and pyruvate carboxykinase. Pyruvate is converted into oxaloacetate in the presence of pyruvate carboxylase. In the reaction, pyruvate is converted into oxaloacetate in the presence of pyruvate carboxylase. This oxaloacetate is carboxylated and phosphorylated by PEP carboxykinase in a reaction driven by hydrolysis of guanosine triphosphate. As you see, oxaloacetate is converted into PEP, and in the same reaction GTP is converted into GDP, and all this reaction is carried out in the presence of carboxykinase. This oxaloacetate is converted into malate by mitochondrial malate dehydrogenase. After the malate is transported across mitochondrial membrane, the reverse reaction is catalyzed by cytoplasmic malate dehydrogenase. In the reaction, oxaloacetate is converted into malate in the presence of malate dehydrogenase, and this is a reversible reaction. After this conversion of fructose 1,6-phosphate to fructose 6-phosphate, the irreversible PFK1 catalyzed reaction in glycolysis is bypassed by fructose 1,6-bisphosphate, which means that fructose 1,6-bisphosphate plus water is converted into fructose 6-phosphate. During this reaction, ATP is not regenerated. Fructose 1,6-bisphosphate is an allosteric enzyme which means that the effectors or ligands bind other than the active site and form the product. The activity of allosteric enzymes are stimulated by citrate and inhibited by AMP and fructose 1,6-bisphosphate. After this what is the formation of glucose from glucose 6-phosphate? Glucose 6-phosphate found in the liver and kidney catalyzes the irreversible hydrolysis of glucose 6-phosphate to form glucose and PI. This glucose 6-phosphate, that is GTP, is converted into glucose. Formation of glucose from glucose 6-phosphate. Glucose 6-phosphate found in the liver and kidney catalyze the irreversible hydrolysis of glucose 6-phosphate to form glucose and PI. The glucose is released into the blood. The glucose 6-phosphate, that is G6P, is converted into glucose. Gluconeogenesis is the energy consuming process. Instead of generating ATP, gluconeogenesis requires the hydrolysis of 6 high energy phosphate bands. After this, gluconeogenesis substrates. What are the gluconeogenesis substrates? There are three important substrates of gluconeogenesis, that are lactate and glycerol, amino acids or alanine. First of all, we discuss about lactate. It is released by red blood cells and other cells that lack mitochondria or have low oxygen concentration. After this, what is Cry cycle? In this cycle, lactate is released by skeletal muscle during exercise. After lactate is transferred into the liver, it is reconverted to pyruvate by lactate dehydrogenase and to glucose by gluconeogenesis. As you see in the diagram, in the Cry cycle, lactate produced by muscle is converted to glucose by liver and feedback to the muscles. After this, glycerol. It is the product of fat metabolism in adipose tissue, is transported to the liver in the blood and then converted to glycerol 3-phosphate kinase. Oxidation of glycerol 3-phosphate to form DHAP occurs when cytoplasmic NAD plus concentration is high, which means that glycerol is converted into glycerol 3-phosphate in the presence of glycerol kinase, and during this ATP is converted into ADP, and after that glycerol 3-phosphate is converted into DHAMP. Amino acids. Amino acids can be converted to glycolytic intermediates, but alanine is perhaps the most important. When exercising muscle produce large quantities of pyruvate, some of these muscles are converted to alanine by transmission of reaction involving. As you see in the reaction, first of all, pyruvate plus glutamate is combined and alanine and alpha ketoglutarate is formed. This is a reversible reaction and it is also called the alanine transmission. Glucose-alanine cycle. What is glucose-alanine cycle? Alanine is formed from pyruvate in muscles. After it is transported to liver, alanine is converted to pyruvate by alanine transminase. Then pyruvate used in the synthesis of glucose because muscles cannot synthesize urea from amino acid nitrogen. The glucose-alanine cycle is used to transfer amino nitrogen to the liver. As you see, first in the liver gluconeogenesis occur and glutamate alanine transminase act upon urea NH3 glutamate alpha ketoglutarate. And muscle protein is converted into amino acids and NH3 glutamate. After that, glutamate alanine transminase and alpha ketoglutarate. Regulation of gluconeogenesis. Changes in the availability of substrate are responsible for most changes in metabolism, either directly or indirectly acting via changes in secretion. Mechanisms that are responsible for regulating the activity of enzymes are changes in the rate of enzyme synthesis, that is in induction and regulation after covalent modification by reversible phosphorylation, and the last one is allosteric effects. Thank you for your attention and time. Presented by Javeria Khan.